So we'll get started with our next keynote for today. Hi everybody, I'm Philip Awadalla. I'm at the University of Toronto in the Ontario Institute for Cancer Research, which is just upstairs. And it's my great pleasure, to introduce our keynote speaker for today, is Janet Rossant. Janet's Professor Emeritus both here at the University of Toronto and at SickKids as well, where she currently has a research lab, and she's the President and Scientific Director of the Gairdner Foundation. She's developmental and stem cell biologist, and has won numerous awards over the years. And I've been told to keep this, and if I tried to read all of those awards, we'd be here for 15 or 20 minutes. We're going to try to keep this very tight. So a great pleasure to have Janet come up and give us a presentation today.
Thanks, Philip. It's a great pleasure to be here. The Human Cell Atlas and all its tentacles is something that is very important, I think, to many people across all areas of biology. And as a developmental biologist, I think obviously the Cell Atlas and the developmental aspects of that is a key component of really trying to understand the complexity of a developing organism. So I'm going to talk to you a little bit today about embryos, but I'm actually going to spend most of the time talking about how a lot of people have decided that maybe embryos are passé, and all you need to do is to make a stem cell model of an embryo, and you don't have to worry about embryos and mice and humans anymore. And the overall message I have for you is that you cannot forget the embryo, and that particularly for people who are undertaking cell atlas work, the fundamental gold standard of everything we need to know is what happens in normal development, in normal tissues, and in the normal embryo. And using that information is going to be critical for being able to assess the new exciting stem cell models. So, stem cell-derived human embryo models, and we're going to look at, are they really close to embryos? And the last part is, well, if they are getting close to embryos, are there ethical concerns that come into play? So we're going to talk about the practical issues of stem cell models and then the long-term ethical concerns. So let's begin and take you back to my favorite stage of development. I work on the mouse, and this is the blastocyst. And the blastocyst is the first stage of development. In the mouse, it's about 4.5 days. Humans, about 6 days. It has 3 cell types and only 3 cell types. The outer layer you see on the outside there is the trophoblast, enclosing a group of cells called the inner cell mass, which by this stage has 2 further cell types, a layer of primitive endoderm on the surface there, and enclosed, the pink cells are the epiblast cells. They're pink because they're stained for a very famous transcription factor, OCT4, which is a pluripotency factor. So those epiblast cells are the pluripotent cells of the embryo, and work from my lab and many other labs over many years, compressed down into 2 seconds. Those 3 cell types give rise to the specific cell lineages in the later embryo, the trophoblast to the trophoblast layers of the placenta, the epiblast to the fetus itself, hence pluripotent, and the primitive endoderm to another supporting tissue called the yolk sac. And what we also know is that by the time you see that blastocyst in the mouse, a lot of experimental data, again, particularly from my lab in this case, has shown that by the time you get to this stage, those cells are committed to lineage. They can't change their mind. Take them out, put them back, tie them in knots, move them around. They know what they're going to give rise to. We also know a lot now about the molecular pathways that specify those cell types, and we know how they arise in the earlier stages. So if we look at mouse pre-implantation development, we know that essentially what I said about the pluripotent cells in the inner cell mass, we call them pluripotent because they're not totipotent. They give rise just to the fetus, but not to the placenta and the yolk sac. So they are pluripotent. What's totipotency mean? Well, the egg, the zygote, is totipotent. It gives rise to everything, all those 3 cell types and everything that stems therefrom. But as we go through development, through the first few days of development, the embryo divides through a process called cleavage, and at the first few divisions, up to the 8-cell stage, all the cells look the same. They can give rise to everything you find, all the 3 cell lineages of the blastocyst. But gradually, by the 16 to 32 cell stage, the outer cells of the trophectoderm start to become formed. You get a polarization, a polarized epithelium enclosing the cells that make the inner cell mass. The inner cell mass itself, by the early blastocyst, is a mosaic of cells that will give rise to the epiblast and primitive endoderm. They sort out, and hence the blastocyst you saw in the previous image. We know it's a gradual process. We know from single cell and other kinds of gene expression analysis that the separation of the lineages is reflected by the general separation of the gene expression profiles, and it correlates with this polarity and position. And as I said, all lineages are restricted by the expanded blastocyst stage. So we know that there are transcription factors, we know the signaling pathways. There's a lot of work really looking at the cellular and molecular interactions leading up to lineage formation in the mouse blastocyst. What we also know is by the time you get to that blastocyst in the mouse, you can actually watch this process now, because we can tag all these important proteins at transcription factors like CDX2, which is a trophectoderm transcription factor. SOX2 is another pluripotency factor. We can tag them using CRISPR with either HALO or GFP, your favorite fluorescent tags, and then do time-lapse imaging. You can watch this process, and you can see that it is indeed a gradual process of separating out the outer cells, the trophectoderm, and only when you get CDX2 restricted to the outside do you now see the pluripotency genes like SOX2 coming up on the inside. So there's a lot to be learned just by looking and studying the cellular processes that are going on. We also know that by the time you get the blastocyst, those lineage-committed cells can be used to derive 3 stem cell lines. The most famous stem cell lines from the mouse blastocyst, and indeed the human blastocyst, are, of course, embryonic stem cells, the pluripotent, hence not totipotent, the pluripotent cells that arise from the epiblast cells of the blastocyst. But my lab showed that you can derive stem cell lines, permanent stem cell lines, from the trophoblast as well. We call them TS cells. And then we can also derive from the primitive endoderm what we call Zen cells. These 3 cell types can be maintained as proliferating stem cells. They require specific growth factors that reflect what the embryo uses to maintain them. Take away the growth factors, they differentiate. And very importantly, this is the mouse. We can actually test the potential of those cells by putting them back in the embryo, what we call a chimera. So if you take embryonic stem cells and put them back into the blastocyst, then what you're seeing here is that the embryonic stem cells give rise to all the cells of the fetus, but not to the yolk sac or the placenta, so they reflect the pluripotency of the epiblast. Trophoblast stem cells contribute to the placenta, not the fetus, and Zen cells contribute to the yolk sac. The genes here that are specific to those cell types are transcription factors that are required to specify the cell types in the embryo. So these become very useful stem cell lines that you can grow in large amounts and study the molecular genetic pathways that specify cell fate. And we've been working on that over many, many years. And I'm not going to talk about the embryo much at all now, because once you have these stem cell lines, then even when we first made these stem cell lines, I would get to this point in the talk, and people would put up their hand and say, well, what happens if you take those 3 stem cell lines and mix them together? Do you make an embryo? Since they come from the embryo, they have that potential. Can you remake an embryo? Can you put the stem cells together and remake an embryo? And we tried various combinations of that, and when you put the cells together, they can sort themselves out and do something that looks a little bit like an embryo. But to be perfectly honest, this is not something that my lab pursued. However, in the last few years, there's been a lot of interest in doing exactly this, trying to ask whether you can reconstitute an embryo-like structure from the stem cells of the blastocyst in the mouse initially, and then later on in the human. And I want to talk a little bit about that today, and how I think it's really important to be able to have some gold standards for understanding whether those stem cell models are really like the embryo. So, can you take those 3 stem cell lines and reconstitute an embryo-like structure? And there have been a variety of attempts doing this in the mouse. The ones that I like best are these. These are called blastoids. This is work by Nicolas Rivron at the Hubrecht lab now, and then in Vienna. And what he did was take ES cells and make a little clump, put them in a small well, and sort of sprinkle trophoblast stem cells on top of them. I liked it, they're our trophoblast stem cells. And when he did that, he was able to show that you could really generate structures here that at first glance look very much like the ones on the right, which are real mouse blastocysts. And he could show that they had trophoblasts, they had the right markers. The inside cells certainly had epiblast markers. Initially, it didn't make much primitive endoderm, but this has been improved recently, and they can get primitive endoderm in here as well. So certainly then, you have this capacity to replicate what looks like a blastocyst. Now, if you take a mouse blastocyst and put it back in the uterus, it'll make a mouse. Do these make a mouse? And the answer is no. So these blastoids, when transferred back to the uterus, will cause what we call an implantation response because they do have trophectoderm, but the inside cells don't seem to be able to go further and make an embryo. It's not quite clear why that is, but it could be that despite there being my favorite stem cells, the stem cells we derive are not quite the right ones with the right capacity to regenerate a functional embryo. And so perhaps what's needed are stem cells that are perhaps earlier in development. So when we met the stem cells from the blastocysts, they somewhat reflect the slightly later stages. Maybe they're not able to reconstitute everything to make a mouse. So we and lots of other people have been interested over the last few years in asking whether instead of just capturing those committed cell types at the blastocyst, could we capture a more totipotent, uncommitted stem cell state? And so what do we mean by that? Well, here we are again. Here's the blastocyst on the right with the 3 stem cell types that I described for you. ES cells, TS cells, and Zen cells, reflecting the lineage commitment in the blastocyst itself. I also told you that during early development, then, we have this progression from totipotency. And I said that by the 8-cell stage, cells were still totipotent. So over the last few years, people have derived stem cells that have seemed to reflect and be able to be maintained in culture. And by various criteria, they believe that they reflect an earlier uncommitted state. These are fairly well-known cells. These are called 2C-like cells that arise in embryonic stem cell cultures. And they express a lot of markers of the 2-cell-like embryo. And it was claimed that when you put them in a chimera, they contributed to the placenta. In our hands, they do not. And nobody has been able to really reproduce that. Also, they are not stable. So this is not a stable state. So we can put the 2C cell-like cells aside. They're not going to help us make embryo models if they're not stable. But these cells are Tu Yang et al., different groups, Pentao Liu and Honghui Deng's lab, in 2017, both published what they called extended or expanded potential cells that they said reflected in gene expression patterns, something like more like the 8-cell stage, and they both claimed that they could get contributions when they put them in chimeras to the trophoblast. Well, my lab was interested in this and skeptical, because we're always skeptical. And it turned out that I was not the only skeptical person. We joined up with Frederick Lanner from the Karolinska Institute, Esther Postfai, who was in my lab and then moved on to Princeton, and Vincent Pasquet at KU Leuven, to ask, well, are these totipotent cells really totipotent? Do they have the gold standards we would require for such a totipotent cell? And we would suggest that a required property of a totipotent stem cell would be the following. Its expression profile should reflect the pre-blastocyst stage, the totipotent stages. The cell lines should be stable genetically and epigenetically, otherwise they're not much use. And if they're totipotent, you should be able to drive their differentiation to generate ES cells and TS cells in vitro. And as an embryologist and a developmental biologist and a mouse biologist, the most important gold standard is that they should be able to contribute robustly to the trophoblast and to the epiblast lineages in chimeras. So while everybody was working on their own research, we decided to reassess the 2 published Deng and Liu extended potential cells against these criteria. And this was published a couple of years ago now in Nature Cell Biology. And I'm not going to show you a lot of data, but I just want to point out that we did look at the expression, re-examine the expression profiles in various ways. This was work that Vincent Pasquet's lab did, and this is just a principal component analysis looking at, again, on the left you have, on the right, rather, you have the phases of early development, and over here is where the extended potential cells from both Yang papers map. And this is bulk RNA-seq, but even if you do single-cell RNA-seq, they move apart for reasons less clear to me, but they still don't move over here into this stage of development. So the expression profiles did not seem to reflect the early embryo. They do make ES cells and TS cells, but not very efficiently. But the most important test is really their in vivo potential. And this is work that my lab did. So if you take normal ES cells and aggregate them with an 8-cell embryo, and if you make them red with a fluorescent marker, or green, as you're going to see in a minute, if you aggregate them together, then they'll make a blastocyst and post-implantation embryos. And if the ES cells are pluripotent, they'll only contribute to the epiblast. If they're totipotent, they should end up out here in what is marked in gray, the extraembryonic ectoderm, EPC, and the placenta. And as a gold standard, again, as a sort of control, what we're looking for a truly totipotent cell, it should behave like a totipotent blastomere. So if you did this experiment, instead of putting ES cells, put an 8-cell blastomere, then you would find that the progeny of that 8-cell blastomere would be throughout these embryos. And that's just shown here. So this was a blastomere from a GFP-positive embryo, and this is the early post-implantation embryo. And you can see GFP cells down here in the epiblast, in the extraembryonic ectoderm, and the EPC. And these are this is an OCT4 to mark the epiblast. TFAP2C is a trophectoderm marker. And again, you see the nice merged images here. So they're really GFP in all the compartments of the embryo itself. But now what happens if you take these presumed totipotent cells? Well, it was quite interesting because actually, they're very good pluripotent stem cells. They make incredibly good contributions to the EPC. So here we're seeing this is from the Lu cells. The Deng cells didn't show any contributions to the extraembryonic regions at all, but the Lu cells did. But you see they're really taking over the epiblast compartment. So if you just want a pluripotent stem cell, I recommend these cells. And we do see GFP cells out here in what is a trophoblast lineage. And you see that blown up here. However, if you look carefully and you look at the expression, the in situ or the antibody staining against L5, which is a trophectoderm marker, and then line it up GFP, you see there's a hole in the ELF expression domain, and that hole reflects where those GFP cells are. So the cells have changed their surface properties, which has allowed them to get out here in the trophectoderm, but they're not trophectoderm, and they continue actually to express the pluripotency genes. So we think that this is really showing us that you have to be very careful in assessing whether you have a totipotent cell or not. The cells are different. Their expression profile is not the same as naive ES cells. And they do have some extent of increased potential to form cyst-like structures, which are claimed to be blastoid-like. But again, without showing you the data, the gene expression profiles of those blastoids is not really reflecting the in vivo trophectoderm. There's still a lot of people working on trying to find a totipotent stem cell in the mouse. There have been a number of more recent papers showing perhaps closer to something that does reflect totipotency. But even if you look in all these papers, the final test of making robust contributions to the extraembryonic lineages is still, I think, needs something to be desired. So it's still not clear whether you can actually generate a truly totipotent stem cell from the mouse embryo. But this is the mouse. What happens now if we want to move this to human? After all, in the mouse, we can work with embryos. We're not prohibited by law or by restricted access. If you're on the human side and you want to study these same stages in early human development, then you have a lot of restrictions, both legal, regulatory, ethical, and just pure access to material. So there's a lot of interest in asking, indeed, whether you could make stem cell models that would give you an unlimited resource of cells for studying human development. So can you use similar approaches, blastoids or various recombination structures, to generate human stem cell models? do we have the right stem cells? So in the mouse, the 3 stem cells, ES cells, TS cells, and Zen cells that people have largely used to put together in various combinations to make stem cell models, do we have them in humans? Well, obviously we have ES cells. They've been derived many years ago. Importantly, what are called naive embryonic stem cells, which are more like the early stages of development, actually have some ability to make trophectodome. They clearly do. I just went through all that data showing you that even people claiming totipotency in the mouse, they are not totipotent. They don't make trophectodome. In the human, I'm pretty convinced that they do. You can take an ES cell, change the conditions, and in fact take a human blastocyst and change the conditions, and you'll be able to generate a trophectodome from the inner cell mass and from the embryonic stem cells. So there is more plasticity. The actual ground state of the human pluripotent stem cell is actually more totipotent and can make trophectodome. And that's an important component that people have used to generate stem cell models. Human TS cells, we tried very hard to generate human TS cells from human blastocysts, and were not able to do so under the same conditions that arise in the mouse, reflecting again that the signaling pathway that the mouse used is not used in the human blastocyst at the same stage. But there have been human TS cells derived by a group in Japan that are more equivalent to slightly later stages, not the blastocyst trophectodome, but they have been able to make TS cells. And there's a couple of different ways of generating Zen cells. We generated them by overexpressing SOT7, and other people have been able to grow them under special culture conditions and get cells that do seem to respond and look like the Zen cells that we see in the mouse. So we've got stem cells. Can we make models? Well, the first approach was to make human blastoids. This is probably the simplest thing you can do. You're just going to try and make a model of the blastocyst. And given that naive ES cells have the capacity to make trophectodome, nobody bothered trying to take ES cells and TS cells and mixing them all up. They just took ES cells and put them in the right culture conditions, put them in little aggregates and small numbers of cells in wells, and were able to generate what appear, again, to look like blastocysts. So there's 4 different papers that came out here. This one is from Jose Polo's lab in Australia. And in this case, he was generating them during the process of reprogramming to make iPS cells. Complicated story, you don't need to understand it. But this is an unusual way of doing it. They kind of caught the cells midstream and tried to capture trophectodome. The other 3 all took naive cells, and in various culture combinations were able to generate structures that do indeed look quite like blastocysts. Perhaps the best ones, again, perhaps not surprisingly, were from Nicolas Riveron's lab, the initiator in the mouse. And here's probably the most like a proper blastocyst. You can see trophectodome markers, primitive endodome, and epiblast in these blastoids. Efficiency, not terribly good. The best methods that have been reported more recently can get up to about 80%. These initial papers, about 20 to 30%, look properly like blastocysts. And you can see, even when you look closely, the cases where they actually were good enough to show you more than one embryo, that there is a lot of variation going on here. So here we are. Here are the structures. I'm the skeptic again. You can make a cyst of epithelial cells that will pump and produce a small clump of ES cells enclosing it, and it's not necessarily a blastocyst. The most important question here is whether those outside cells are actually trophectodome. Is this really a blastocyst model, or is it just a cyst-like differentiating embryonic stem cell? So again, how do you assess the validity of these human models? We can't test the in vivo potential. I can't make a chimera in the human, so the gold standard is missing. So there's a considerable reliance from all groups and all studies in this on comparison of gene expression to the intact human embryo. And so that becomes the gold standard. And then you have to have very good expression data from these early stages of human development. So I sort of re-established the grouping to look at how to assess this. And Frederick Lanner's lab, who we'd collaborated with on the mouse work, has gone on, in particular Chen Zhao in his lab, has been really trying to develop what they call a sort of reference tool to look at the trajectory of gene expression, single-cell gene expression analysis in human embryos, pulling data from a number of different published studies from pre-implantation right through to the Tizer et al. Study, which actually was a study on a very rare early human gastrula that was obtained from termination material. So this gives you a trajectory from the early embryo right through to gastrulation. And you can just see that you start up here with 8-cell, morular, pre-lineage, that's the sort of human specialty, inner cell mass. Then you go out here to the trophectoderm and its differentiated progeny. Primitive endoderm goes down here to the endoderm, and then the epiplast goes down here to form mesoderm, amnion, et cetera, et cetera. So this is the trajectory, and they've been working on this and revising it, and this is actually not yet published. This final trajectory is unpublished. You'll find an earlier version in that bioRxiv paper that is up there. So then they've been testing this out, and just, does it work? So first of all, you want to know whether normal stem cells map where they're supposed to map, and they do. So the naive cells, the naive embryonic stem cells, map close to the inner cell mass, which is what you'd want. Primed cells, which is the sort of somewhat more differentiated embryonic stem cells, map down to the post-implantation epiblast. Those extended potential cells, which were also derived in human, as they do in the mouse, they map to the epiblast. They don't map to the pre-implantation stages. And then the Okai trophoblast stem cells, luckily, also map to trophectoderm. So it looks good. Then you can ask, and that's what Lano et al. Have done, where do the blastoids map? How do they look? And this is what you see. And essentially, they're not bad. If you look at these three, you can see that there are cells where you would hope to see them in the sort of epiblast going down here to the differentiating mesoderm, over here in the trophectoderm, and in the primitive endoderm. In all cases, not much primitive endoderm in this one, but there is trophoblast, there is epiblast. Yanagida also, you see them going down 3 lineages. This one, which is the eye blastoids, you really don't see many cells mapping over the trophectoderm, and there's a big blob of what they annotated in the paper as trophectoderm are actually mapping where the amnion maps. You'll also see, and this is typical of all these stem cell models, that there's a lot of noise. There's a lot of cells that map all over the place. There are clearly other undefined cells in all these structures. And I would note that in almost all these cases, in fact, all of them, before they did the single cell analysis, they carefully selected the best looking blastoids they have. So if you were to take the overall variance, there's a hell of a lot of variation going on. But, some of them are getting there. There's no question about it. So this amnion, the fact that the eye blastoids have amnion instead of trophoblast, this is a bit of an aside, it's a sort of developmental human problem, that amnion and trophoblast turn out to be a problem in separating in the human embryo. There's a lot of conflicting data about where the amnion comes from, can you get it from stem cells? And Rugg-Gunn's group in Cambridge has actually shown that in the normal human embryo, there's 2 phases of making the amnion. And the first phase, the cells actually turn on markers like trophoblasts because they have properties that are similar to trophoblasts. But then later on, they differentiate into proper amnion. So it just means that whenever people are trying to really interpret lineage identity in these stem cell models using single cell expression profiles, you have to be very, very careful. And the initial annotation of your gold standard trajectory is important, but you have to be very careful that when you're annotating a cell as amnion versus trophoblast, that you use not just 1 or 2 markers, but that you really use the full trajectory of the model. So stem cell models then, these blastoids are clearly not identical to embryos. The epigenetics, it's not surprising if you think about it. These are all derived from stem cells that have been cultured for many generations in the petri dish. It's amazing they do anything at all. And clearly, the epigenetic state of those stem cells is imperfect. We know that stem cells, some of the imprinting differences and so on that occur during normal development become defective during stem cell cultures. Also, it's very unclear what you're starting with in many of these cases. All the different groups use different protocols, different starting cell lines, different differentiation protocols. It's very unclear. A lot of variation. It's also, if you read the papers carefully, in many cases, it only works with one particular stem cell and not another. So there's a lot, this is not a robust system. And it's not a replacement for studying the embryos themselves. They can be useful. I've not talked about later embryo models like gastruloids and somatoids and all the various organoids that you've all heard about. And they can be very, very useful because they can be used to study specific limited events. But if you actually want to mimic the whole process of development, you really would like, I suppose, in the end, to make a functional embryo. So can you ever do that? Well, you can't test it in the human, but you can in a non-human primate model. And this is a recent paper from Zhen Lu's lab in which they actually did what had been done in the mouse as well, and again, go back to this concept of having the 3 blastocyst cell lines, and they generated TS cells, N cells, and ES cells in various ways in a monkey system, put them together, made a blastoid, allowed it to differentiate in culture, and were able to get structures that look like the early post-implantation monkey embryo. They did single-cell RNA-seq, and they could see some of the components of the early post-implantation stages. So this suggested this is a good model for these early stages. Then they transferred it back into a pregnant monkey, and I don't know if you want what answer you want. The answer is that they were able to implant and cause an implantation response. But again, like in the mouse, these did not go any further. So this system, although it looks quite good, even in this rather well-defined system in the monkey, there's something missing in these stem cell models. So they are good for studying some of the phases of early post-implantation stages, but they're not replicating and producing an entire embryo. There's been a recent set of papers, and there's more lurking out there. There's a lot of noise in the system at the moment on generating more complex human stem cell-derived embryo models, similar to what I just showed you in the cymologous monkey. Again, not just taking ES cells and making blastoids, but taking ES cells, differentiating them in various ways into ES, then, and TS type equivalents, and then combining them and putting them into more complex culture systems to try to replicate the post-implantation stages, actually without making a blastocyst first. And these are papers that you can find online, and they all, in different ways, were able to generate structures that were able to mimic some of the early events of the early post-implantation human embryo. None of them were perfect. All of them were very low efficiency. In other words, you put these things together, and mostly what you get is disorganization. But in some cases, they were able to generate structures that did have information similar to a human embryo. And I'll just show you one of these to make the point that if you're going to generate a stem cell model, it's not enough to put cells together, grow something that looks like a blob of differentiating cells, do single-cell RNA-seq and say, you know what, my blob of aggregated cells contains all these cell lineages because I can see them in my single-cell RNA-seq. We've known for a very long time that you can take embryonic stem cells and generate what we call embryoid bodies, which is just you grow them in culture, you put them in suspension, and they differentiate, and they'll make blood and heart and all sorts of things, but they are not generating the organized structure of an embryo. So they have to make an organized embryo. And those papers, in various ways, show some degree of organization. And this is taken from the paper from Jakob Hanna's lab at the Weizmann Institute. And this is what you should be looking for. So it should make something look like a blastocyst. It should have the epiblast, the yolk sac, the amniotic sac. And this is diagram taken from sections of real human embryos. And you see that the models are lining up quite nicely against them. And then if you look at gene expressions typical of the lineages, they also line up with the embryos involved. So this then is the best case scenario, but again, his percentage success was 2.9% of the original structures. And now, what happens if you take those structures and put them onto the Frederick Launer and Chang Zhao's profile? And for reasons best known to themselves, they turned this upside down. So this is taken from the paper from Jakob Hanna. So they collaborated with Frederick's lab. And now the trajectory, the reference trajectory, is upside down from the one I showed you earlier. So we're starting down here with the 8-cell and going up here to trophoblast, hyperblast, and epiblast. But here's the data from the, again, selected embryos from the Jakob Hanna's paper. And this is the kind of thing you would hope to see, and it's not bad, right? So you're seeing epiblast cells, extra-embryonic mesoderm, you're seeing amnion, which you'd hope to see this time, the post-implantation stages, yolk sac, and trophoblast. Not a lot of trophoblast, mostly because they were getting syncytial trophoblast, which doesn't dissociate well. So essentially, this is the kind of data that you would hope to see. So things are moving. There's a lot of activity in this area. It's very important to be able to have careful gold standards of what you mean by an embryo model. And none of these are anywhere near a real embryo. But, you know, what if? And there's a lot of people saying, well, what if you actually do this in human and you get better and better at it? What happens if those integrated stem cell models, like the one I just showed you, cross the boundary and become close to real embryos? Do we have ethical concerns about generating human embryos in a petri dish, and what are the regulatory concerns around that? And it's complicated, and it's another whole lecture, but just to say that the International Society for Stem Cell Research takes all these things very seriously, and we have a set of stem cell guidelines which we revised in 2021, and 2021 ahead of many of these papers, but you know, we know what's coming, or we think we know what's coming. And these are some of the guidelines that are in there about this kind of work. None of these stem cell models should ever be transplanted to a human or animal uterus. These integrated models that include trophoblast and primitive endodome potentially could make a whole embryo, should be really subject to very careful review. And if you're going to culture them, either an embryo or a model, you have to do it for a short period of time. These are in vitro models only and should be considered that way. There's something called the 14-day rule, which applies to human embryo research, especially here in Canada and around the world. It says you can't culture a human embryo beyond 14 days. And if you were to do this with these stem cell models, the same thing would apply. If you want to change that, there's got to be a full public engagement. So there we have it. You can go from study an embryo. You need to understand the lineages of the embryo, and we still don't know everything there is to know about how an embryo forms, especially in the human case. We don't fully understand the relationships between the embryo and the different stem cell states. Cell atlases are going to be essential tools for standardization of these human stem cell embryo models. And it all begins and ends with my favorite stage of development, the blastocyst. And I'll stop there and thank the people who've been involved in some of these later collaborations in my lab, the chimeras, and the other people.
Great, this is on. Thank you very much. That was a wonderful presentation, Janet. So I'd like to open the floor for questions. And when you have a question, please introduce yourself and tell us where you're from. And also that opens up to online. I'll give you my mic.
She's got the mic.
Mic. Okay, great.
Hi, this is for the online. This is from Donna at Sloan Kettering Institute. Do you think SC-ATAC-seq might be more informative of cell lineages, cell potential, and plasticity, and a better way to try to compare the different systems you're looking at?
So the question is, I only talked about gene expression profiling, and obviously here today everything about multi-omics, and certainly ATAC-seq is being done on the early embryo, as you know, and it is something that I think would be very useful as a comparator. And in fact, a lot of people are trying to do that. But I think the stem cell models are sort of just beginning to be developed. And so at this point, most of them are just doing expression profiling. But some groups are starting to combine that with a TAC-seq. And yes, it probably is a better way.
Right here in the front.
Great talk. It's a question for speculation, I believe. If you had to make a guess, is it the signaling or the physicality of in vitro versus, what do you think gives in not making these? Is it really, you had to.
So the question is really what's missing, even when you've got something that looks pretty good. I mean, again, they're not all doing it, but the best ones look pretty good. So why don't they go further? I'll speculate, but I don't know. I think one of the things is that there's, particularly in the human, there's just a lot of variation between cell lines. There's not enough standardization of the starting material. And if you're thinking about the starting material has to have the full pluripotency that makes everything. Any small genetic, and there are genetic, but epigenetic changes that occur in those cell lines are going to be, they'll start off looking okay, but they're not going to make it through. In the mouse, it doesn't quite, I'll tell you why that's probably not such a good speculation, because in the mouse, we know that we can take mouse embryonic stem cells and make a mouse. This is an experiment my colleague Andres Naj and I did years ago, where you can take mouse embryonic stem cells being grown in culture, aggregate them to a tetraploid embryo, and you can get live mice. And yet, again, those cells have been in culture a long time. Do we culture them better in the mouse? Maybe. But again, I don't know, so I don't know. But it does mean that I don't think there's any intrinsic reason why it's not possible, because certainly in the mouse, you can make a mouse from stem cells.
In a similar vein, is there a question of efficiency, which you brought up many times? Is it related in some ways to the same thing, that we actually don't know what the reasons are, so the variation is so huge? Like percentage of 2% even look good. Is it also sort of in the same direction, that what the starting material, we don't know what it is? So.
Maybe. I just think it's very early days for all of these systems, basically.
Thank you so much. It was such a nice talk, and thank you for sharing such a long history of research with us. My question is regarding the health of the surrogate uterus. How long do you keep the mice after, you implant the cells in their uterus? Have you ever looked if they develop any further, I don't know, tumorous structure in the uterus afterwards? Because you said, it was not pretty successful. You went up to a certain level, but then it failed.
When people put the blastoids back in the mouse, they would, the mice would be sacrificed a few days after implantation, so they don't go further. And they really don't make tumors. They just really basically seem to die.
It's short term, They don't know what it does in long term.
No,
so that's also something to consider.
No, they don't make chimeras. They don't make tumors. So embryonic stem cells, when we put them back in chimeras, in the early days, not embryonic stem cells, or what were called teratocarcinoma stem cells, have this ability to be pluripotent and make chimeras, but they would still have this capacity to make tumors, and the mice would be born with tumors. That was not a good story. But embryonic stem cells, although I said there's variation and they may not be perfect, they generally speaking don't make tumors.
Thank you, Jen. I just have, I guess, one question is, so should we still do a stem cell atlas? Wouldn't it be useful regardless how far we get?
Well, I think that clearly there's a lot of need for standardization across everything in this early embryo and stem cell area. Part of the standardization would involve cell atlases. The ISSCR just published a set of guidelines about if you work with a human embryonic stem cell and you want to publish a paper, you're supposed to be able to put down how the cells were cultured, what the genetics is, what the it's horrible. It's a lot of work. I think people are going to have a hard time doing it. But in essence, that's what's missing from this field, is that kind of consistency. So until we can get that consistency, I'm not sure that you'll be able to get a really good stem cell atlas. And I think the consistency has to come before you necessarily. I mean, you can do the analysis, but you really want to define the system better.
Okay, that's a great message. And I'm afraid that's our last question today. So thank you again, Janet. That was a wonderful presentation.
Hello, everyone. I'm Sergio Triana from the Broad Institute, and I'll be introducing the Lighty session. And in that note, I'll introduce Alex Tong from Myla Institute and the University of Montreal, who's going to talk to us about perspective learning continuous dynamics from time-lapse single-cell data.
I'm Alex. I'm at Mila, and I do machine learning for single cell, and I do modeling of dynamics. And so what does this mean? Okay, so we have this problem where we collect a lot of single cell data, and it's very plentiful, but it's sort of over time, and we have very discrete time points and just distributions. And so how do we learn sort of cell dynamics from this type of data where we just have distributions over time, but no knowledge of the exact dynamics? And so the idea here is basically we're going to do the machine learning task, which is to say we're going to leave out a time point and try and interpolate and find a distribution that matches that time point. So we want to learn a model that gives the sort of same distribution of cells the best as possible. And so the reason that you might want to do this is because if you can learn how cells change over time, then you can do many other biological tasks that are not necessarily just imputation, but are sort of more important biologically. And so the general idea here is we're going to learn a dynamics model that predicts from one cell and at the time what the change of the cell should be. And we're going to use integration techniques to integrate that cell over time and find where it goes sort of from time 1 to time 2, or where it might be at a previous time. And so this is a very powerful method to sort of integrate all we know about cells that are from the sort of cell state to the change in cell state. And so the idea here is we have single cells over time, and we're going to learn this function that will transport the mass of cells, but only supervised by where the populations are, rather than sort of the individual trajectories of cells. So this is a single cell time series of actually embryo bodies, and so over 30 days, and you can see how the cells move over time and match the distributions. And so these models are based on neural ODEs. So this is a machine learning model where you predict the derivative cell states. And there's many ways. What's very nice about this is there's many ways we can bias these models to match our priors about the data. So a lot of the priors we use are optimal transport based. So we know that cells sort of move in straight-ish lines in some cost function. We also know that cells move along a manifold, so we match density. And then there's lots of methods on RNA velocity that sort of can measure instantaneous change. So we can also have supervision of the instantaneous change at a bunch of places. And then finally, we can use what we know about cell proliferation to regularize the growth of cells. And so there's many different ways you can do this. One of the ways we've done recently is to sort of learn a latent space where it's a geodesic distance, which happens to work very well in the cells because we sort of have all these UMAP-based methods and t-SNE and all these things that are based on graphs and nearest neighbor similarity. So preserving these distances helps a lot in the interpolation. Cool. So what can you actually do with these? So I'm going to present two methods, two ideas. So one thing you could do is try to improve the yield of in vitro models. So one of the problems we have is we have 3D cell cultures, and only 10% of the cells you seed actually develop in tumor spheres. But you'd like to increase this yield. And so we have sort of a cancer stem cell population which differentiates, but we would like to know, yes, these cells differentiated, what sort of markers were they most likely to come from? And so we scored our model's estimation of proliferation rate with a bunch of genes and found that these two genes in particular, cell surface markers, were very good, had very high correlation with the model's prediction of what type of cells would proliferate. And so we also can look at these and sort for them and find that, yes, indeed, these two markers are very useful for increasing the yield. So we can actually increase the yield 1.7 times by sorting for these two markers. Second application is to look at gene regulatory networks. So this is a very hard problem, and I'm not at all suggesting that it's solved, and this even comes close. But the idea here is that we learn the change in genes over time. We also sort by known gene regulatory networks, and then we find a sort of subset that we're interested in. So this is looking at MET, so mesenchymal to epithelial transition, and we try and find the genes that regulate this transition. And so we found a couple genes that seemed upstream of this process, and when knocked out, they control the process. So this, I think, is an interesting application of learning these temporal networks to sort of untangle the gene regulatory structure. Cool. And so just the last thing I want to talk about is, I think, very exciting. So these machine learning models have worked pretty well, but they're sort of hard to adopt because they're very slow to train and it requires a lot of expertise. But there's sort of these new models that are based on diffusion, and it's really way easier to train. So these are like 100 times faster to train, basically. And you can, so this is not developed just by us, but there are many other people doing similar things. But you can essentially condition on both the source and the targets and match distributions over time. And so this sort of simulation-free method is very fast. And so it improves this neural optimal transport performance. So this is one of the common tasks to use optimal transport in these methods. And so the performance is much higher, in this case, over the previous methods where we sort of had to integrate over time. And furthermore, you can add stochasticity. So this is one of the problems with our previous methods. You could do ODEs, but not SDEs. And this way, you can do SDEs with very little additional cost. We call this simulation-free score and flow matching. And finally, this yields like a very high performance on the sort of left out time course tasks. So you leave out a time point, and in 1,000 dimensions, you see how close the distribution is in sort of this earth move resistance. And so this performs very well. So it scales to high dimensions where we could only do sort of low dimensions where we're doing the interpolation. So I just want to sort of takeaways, right? We have this sort of method where we still learn this transition over time, but now if we learn this with simulation-free methods, we can learn it much faster. Yes, thank you very much. And yeah, I'd like to thank all my colleagues.
Thank you, Alex. We have time for around 1 to 2 questions. We can start with the virtual participants, if there's any questions. Nope. Any questions from the audience?
Hi, thank you. That was very nice, and I think you're touching on something that is really useful and really needed. Now, my question is, what do you do next? And what I mean is, if I understand correctly, you're starting from in vitro models, you're then creating your computational model. So how do we, I think the important bit is then go into the tissue and see if these trajectories exist.
Yes. Yes. So these models currently are trained on just one data set at a time, right? Because we don't have enough information to transfer to, say, a different system, right? In vitro to in vivo. But we would like to ideally train on many data sets at once and sort of make this transfer. So yeah, we need more data to make this work well. So it's sort of the start of these models. I think we can use sort of the genes, right? The top level features that you extract from it might be useful suggestions of what could be useful, but it's currently you can't just apply it to in vivo modeling.
So how important do you think is to start the model with also your in vivo data, so rather than just the in vitro?
Yes, it would be very important if you have this data already. Yes.
Okay, thank you.
I have a question. I want to know how many time points should we prepare to use this method? Maybe it's better to prepare a lot of time points, but we need more cost to sequencing.
Yes. Yeah. So I think the rule of thumb that I use is if it overlaps in the UMAP, right, then it's easy, and there are transitions that you can look at, right? I think if you have long stretches that are missing, it's much more challenging and you have much less certainty about what's happening in between. So, it's, you don't know, but yes, at least a little bit of overlap is key.
Thank you.
Thank you, Alex.
Thanks.
I would like to introduce now to Romain Lopez from Stanford University, who's going to talk to us about causal modeling of single-cell perturbation data. Thank you.
Hi, everyone. My name is Romain. I'm a postdoc at Genentech and Stanford. I would like to show you this green sticker. This does not mean that I was here at the first HCA meeting. This means that this is my first time being here, and I'm really happy to be here for the first time and be part of this community. So thank you very much for the organizers. Now we can start my talk. So today I would like to talk about perturbation data, and I want to highlight that perturbation data informs us on cause and effects. For example, we put drugs in cell and we see how they react. I'm going to talk about the collaboration with Calvin Chen from Osaka University. Here we're studying the effect of drugs on T-cell activation. So for example, we take cells from human, cells from mouse, we isolate primary CD4 T cells, and we can do an array screen with tens of kinase inhibitors with different dosages, and with an in vitro model of T-cell stimulation, anti-CD3, anti-CD28, where we wait for 72 hours and we're able to profile intracellular protein staining, so looking at phosphorylation of antibodies, but also surface protein and RNA sequencing. So this is a nice use case from which we can try to understand what are the biological questions we can answer from perturbation data. So for example, how do drugs affect T-cell activation? Do drugs act on their documented target? Whenever you buy some of those small molecules, they're usually documented. Then, are there differential effects occurring at high versus low dosages, or drastically different effects? We'll see an example of that. And which effects are conserved or not between mouse and human? I won't talk about this today, but it's there and it's interesting. So I want to highlight that most of the things that I've been working in the past that are probabilistic models, these models are not causal. So causal meaning that we could apply them to perturbation data, but they don't have a right semantic or the right framework for thinking about it. So during my PhD, I worked on single-cell variational inference, ScanVI, and the SCVI tool software. And in a few sentences, basically, we model gene expression as a function of batch effects and cell states that you can see in most of UMAPs that some of them that we've been seeing at the conference. And cell type can be observed or not, and we can use that for stratifying cells into latent space. We've been seeing a lot of those cell type labeling, batch effect correction, and the reason for which I really like this framework is that it's really easy to extend to multiple modalities. It's pretty straightforward, actually, and it scales to millions of cells. So, and for example, a lot of the code used in this publication uses some of this, it was used for analyzing the HCLA data. So now how do we go from this previous type of model I showed to what I call causal model? So causal models must specify strict rules about what the perturbations do, and that's to me, that's the only difference between a non-causal and a causal model. So what are examples of this type of models? For example, one of them is causal structural learning. In causal structural learning, I'm going to talk about this as a gene-centric model. Basically, we think about genes as regulating each other, and we have those Bayesian networks that specify how expression of genes depend on expression of parent genes. And here, if we had genes knockout, which is not what I'm talking about today, we could basically be able to model interventions that break in correlation from each gene to its parents. And in that specific setting, I did some early work in my postdoc that I want to keep working on, but that's not what I will talk about today. I would gladly talk about it at the coffee break, on how to factorize those gene networks to scale up inferences and to apply this to real Perturb-seq data. And there are quite a lot of interesting challenges there. The reason for which it's not very suitable for chemical screen is we don't know what the target is. There could be several targets. It's extremely complicated. So another way to think about it is to think about cell-centric causal model. So, and this is linked to what's called causal representation learning. In this kind of model, we go back to kind of SCVI or things I've presented before, but here we think about, again, gene expression depends on latent states or latent variables, but the perturbation are actually changing those latent variables. And so one example, Fabian, for example, had some work on this a few years ago. So this is not a new idea, but when you start thinking about it in this perspective, you can have other ideas. For example, because we're perturbing latent variables, how about we perturb a subspace of latent variables? What does it mean? What do we gain as insight from this type of models? And we can think about a lot of other inferential questions. For example, is there a subset of latent variable that's affected upon perturbation? And I've did some work on this as well, but that's not what I'm presenting today. If we have this kind of sparse subset, we can think about what are downstream genomic features affected by those perturbations. We can think about, if we go back to the drugs, we can understand how drugs relate to each other based on how latent variables are changed. And if we think about subspace intervention, that's what I want to focus on. How do we separate variants, or how do we disentangle the result of those perturbation experiments? So let's go through an example. Here is a model that I'm interested in for today. So we have Z that is a subspace of latent variables that explain the omic variation present across all of my vehicle cells, my non-targeting control. I mean, it's not a CRISPR screen, but my control cells. S will be the omic variation induced by small molecules. So I'm going to activate new sources of variation, and I want to know what those are, and that will be targeted explicitly by the small molecules. Now we have gene expression measurements. We also measure surface protein, as I said, intracellular protein measurements. So what can we do with this type of model? First, because we're able to separate these variants, we're able to really tease out the effect of the perturbation. And if we just look into the salient space, so this S, we're able to separate drugs much better, and we're also able to cluster drugs by affected pathway much better. But that's a bit of a superficial type of analysis, but it's important. And then what's more interesting is what kind of biological processes are present in the background, the Z space and the S space. So I can't explain everything because I only have 30 seconds left, but I'm going to basically tell you that in my Z space, the way I think about it, I want to put in there the omic variation present across vehicle cells. And indeed, I can use hotspot to kind of get an idea of what are the gene modules present in that latent space. And I can go back to the user gene module to get gene sets and enrichments. And we see a lot of cell cycle, but because it's T cells, we actually see a lot of other things like metabolism, oxidative phosphorylation. And we can reproject the drugs and calculate the activity of those gene modules. And for example, the biggest point of the heat map is this, which is the TBHQ, a small molecule, and we look at gene module 1. Gene module 1 is very specifically enriched for a lot of things, but including oxidative phosphorylation. And actually, TBHQ is an OXPHOS inhibitor. So that was kind of an interesting thing, that we cannot just remove this background variation in vehicle cells because it does convey some biological information. And then my last slide, in the S, we can basically run a more classical differential expression by controlling for the background, and that's actually quite a bit technical, I can't talk about it. But we have a lot of gene programs. When we cluster this heat map, those genes programs, some of them pertain to, of course, T cell and calcium signaling and a lot of other things. But if I look into, I like this one, gene program 5, because if you look at those JAK-STAT inhibitors, you can see that at low dosage, you have upregulation, at high dosage, you have downregulation. And actually, in those genes, you have apoptosis and BAX. And it has been documented already in cancer cell that some of those JAK-STAT inhibitors have very different mechanics at like low and high dosages. All right, so in conclusion, there are many potential ways of modeling perturbation data. I'm not claiming that this is the best way, but it's definitely an area that's really exciting to explore. Causal machine learning can provide insight on and new ideas on how to make algorithms and how to think about those models. I've presented this algorithm called multi-contrastive VI, which is an extension of previous work called contrastive VI that just got accepted, and it's a really exciting work that you should check out. And I want to finish by saying that the data we have is not exactly ready for like the causal inference that we'd like to make, to think about causality, but we can still get insight from this machine learning community. And I hope that provide insight on how to engineer the data better for making better inferences. So if you would like to talk about this with me later, I'd love to. Thank you very much.
Thank you. Any questions from the audience or from, thank you, the gentleman. Please introduce yourself when
Certainly, thank you. Great talk, Romain. My name is Danilo Bzdok. I'm a scientist at Celerity. I'm interested how you think about the fact that your underlying mechanistic model is in a single cell, but you have to destructively sample a population of cells to get the measurements that we're looking at here. How do you propagate that information up from individual entities out to populational distributions?
So actually, the way that the model works is the graphical model specifies the gene expression of a single cell. And for example, when you think about these models where, and that's precisely why when you think about these models where some part of the subspace is invariant to the perturbation, if you have batch effects, for example, the model will put the batch effect in the background. So it's able to capture, yeah, it's definitely a model for a single cell that just aggregates information across the perturbation. And the way you do this is just decomposing the likelihood, and it's all just calculating the likelihood you're interested in. And the guides are IID, all the perturbations are IID, everything decomposes, and you can still apply a lot of classical methods for machine learning in that case. Yeah.
Thank you.
The lady in white.
Hi, I'm Michela Nozeda from Imperial College. Really great work. My question is a bit generic, and you have used the chemical screening, and I'm curious why you chose to go with the chemical screening and not doing a genetic screen knockdown. I'm not saying it's wrong, I'm just curious of why you took that approach where a drug might have multiple targets, right, instead of doing a knockdown, for example.
So I'm also have other projects on which I'm working. So this is a great question. The reason I'm presenting this work today is because it's the most presentable. I'm also working on perturbation data, like genetic perturbations. But first, what's kind of interesting is that with chemicals, actually the effect size is larger. So for thinking about this subspace disentanglement, this is a much, I mean, the model is more appropriate to the data, and the data is more appropriate to the model, and that's why I'm talking about this today. For thinking about causality in more general fashion, potentially the perturb-seq is more important, but again, it depends what framework that you're thinking about. And I think that, yeah, for what I've presented, actually, this is one of the most ideal data set I could have.
Thank you.
Thank you, Romain.
Thank you very much.
Next. Next, we will have Charlotte Bunne from ETH Zurich, who will present us about neural optimal transfer for single-cell biology.
Thanks a lot for having me. So this will be some elements of both talks, and I'm going to... Yes. So we are studying single-cell perturbations, where, in particular for personalized medicine and the observational clinical cohort study I will briefly mention at the very end of the talk, we would like to understand how does a patient really respond on the single cell level to a treatment. And so often we have provided single cell data of a tumor before and potentially after, in vitro after a treatment, and we would like to understand on the level of single cells, how do these cells respond to a perturbation. And of course, with the revolution of the omics, we have now high throughput methods or high throughput data available, which make the use of machine learning actually feasible and come in to answer questions such as, how is a patient actually going to respond to a drug? What is the most effective drug I could potentially choose? And what are particular biomarkers of a disease? So what we still see is that traditional methods of machine learning not necessarily excel in this regime. And this is due to very particular factors which come with the data we are provided with. So as you all know, single-cell technologies, omics, are destructive. So every time I measure a cell, I actually need to destroy it. So what I'm interested in here, the dynamic response of a patient to a drug, is nothing I can actually measure. I can only measure the cell population before or after. And I somehow need to fill in the gap. So the classical setup we have in machine learning of paired data, an observation before and a label, is something we are not provided with, and we need to get creative. Similarly, despite the upcoming rise of spatial transcriptomics, of course, we also lose not only the temporal but the spatial context, and we are often provided with relatively low sample sizes. So we need to design new machine learning models which somewhat are tailored to the single-cell biology and use the inductive bias of the process as core elements of these algorithms. And one idea here is, and one example I will present, is the use of optimal transport. And again, the setup is we have potentially a cohort, single-cell profiles of several patients, and now we would like to learn, like find a neural network which predicts how a population of cells responds to a drug. And we want to learn this because if a new patient comes into the clinic, we want to use this knowledge we have, these trained networks, and predict for an unseen patient how they would respond. So again, as I mentioned, the problem is measuring requires destroying. I cannot measure the same cell before and after. I only have access to samples of a distribution of unperturbed and perturbed cells. And of course, I could just look at the average effect, but we all know that in particular in cancer, tumor heterogeneity is a driving factor of understanding resistance mechanisms and so on and so forth. So I want to do these predictions on the level of single cells. So one thing we need to do is we need to somewhat reconstruct this alignment between these unaligned distributions. And the question is, what is the most optimal T to choose from? So I think all my math notation is a bit corrupted, so let's see when we reach the equation. So we need to somehow realign and find a matching. And the question is, what is the optimal matching? Like, what is a good inductive bias here? And one educated guess we can make is to use, basically to find the most similar cell in the perturbed population. Meaning, I find an overall matching which reduces the overall optimal transport cost. Well, there's an integral missing. So basically, I want to find the overall matching which minimizes the misalignment cost. And this is a principle which coincides with the theory of optimal transport, a very old field of mathematics, which has been previously used in single-cell biology to actually find descendant cells. So here, of course, we want to use this and parameterize this optimal transport with a neural network. Because if a new patient comes into the clinic, we want to make predictions and predict how this cell, how this patient would respond to a treatment. So we are going to use some theoretical properties we are provided with from optimal transport, in particular the fact that we can parameterize this optimal transport map, this Morse map, via the gradient of a convex function. And for this, we can basically constrain the class of neural networks we want to parameterize to those which are not, like parameterize the family of convex functions. And that's a quite popular approach now in this neural optimal transport field and something which has been picked up in many different works by now. So, of course, we know that if we have a patient, the response to a treatment is not solely determined by the single-cell profiles, but there's, of course, a lot of meta-information which are going to influence something we not necessarily measure in our single-cell experiments. So we provided a recent generalization of the approach to be able to condition such optimal transport, which describes the response of these tumor cells on the single-cell level, to condition it on extra meta-information, patient meta-information we are provided with. And so this is currently part of two papers, one published at Nature Methods and the other one in a machine learning conference called NeurIPS. And initially we started and tested our setup on a system of melanoma cell lines where we know the treatment outcomes. And now, once we knew that this is really providing us very accurate results on the single cell level, we moved to an observational clinical cohort study where we have 47 patients, 47 melanoma patients, and we have screens for 35 different cancer drugs. And so this is going to be a study which hopefully will be public in September. I would like to briefly set this optimal transport a bit into perspective and also highlight some other works of mine over the last years. So optimal transport is not just a matching, right? It's a very rich mathematical field which combines actually control, like it has links to control theory and dynamical systems. So of course, primarily it gives us maps, and they allow us to study the response to perturbations, but it also has very intricate links to the theory of partial differential equations, and there's some works where we have neural networks which basically describe now for multiple time points, similar to what Alex was mentioning at the very beginning, the evolution of single-cell populations when we are provided with several measurements. And even now, and this is really the link to diffusion models, we have the link of optimal transport to something which is known as control theory. And that is becoming very exciting because we can say, In control theory, I have an active strategy and I want to steer my cellular system into a certain direction. Can I find paths in the cell space which steer us to a healthy state? And so here we have also very nice links to those so-called diffusion models, a very powerful class of neural networks. And with the power of optimal transport combined with neural networks, we can really learn very interesting cellular dynamics over time. And so then what are the next steps, of course, to learn these like space-time processes from multiple snapshots. And this is something I have been doing in previous works by using this neural parameterization of partial and stochastic differential equations. And my time is up, and here are my collaborators, and thank you a lot for the time.
Thank you. Any questions from the? Nope. Any questions from the audience? The person in the first row, please.
Thank you very much. So you made it very clear how important having longitudinal data from the same patient was, and the problems associated with that as well. But I was wondering, in the context of, say, moving this forward to when a patient walks into a clinic, you may not have access to that longitudinal data. There's plenty of potential cross-sectional data from multiple patients. Have you tried thinking about how to move into that space, or tried your algorithms in that context as well?
So the later models is actually trained across several patients, which comes with their own set of challenges. So there we actually try to learn like somehow a coherent model across different patients. And then if I have less data or only like an initial snapshot of a patient provided, then I can predict the response to a patient. So in that sense, we are already trying to combine different patient data into a single model.
Great, thanks.
Actually combining between the two consecutive talks,
yes.
Have you thought about that field potential and to combine it with perturbation data to try and find the cause of the transport, essentially?
Yes, yes. So I tried basically this conditional model. I tried to basically condition it on the action I take, on the perturbation I apply. I think the core element here is that you need to have some good representation of your perturbational space, and I think that becomes more or less a research question on its own.
One.
Yeah. But so you can somehow think of this conditional OT model a bit as a causal effect model. Basically, you want to say, okay, I want to predict my distributional shift given that I have a certain somehow intervention, right? And so then the question is like, how can I model the dependence on the intervention? The model parameterization here I used, and this also has a part in the paper, is actually conditioning them on actions. So we have a perturb-seq data set, and I want to condition on the genetic perturbation I apply. It very much becomes a problem of representing your perturbations well. Yeah.
Thanks.
Thank you, Charlotte, and thank you all the speakers from the session.
Okay, everybody, we have a 15-minute break, and then we'll be back here in this room for the next session. Thank you.
Hello, welcome back to session 11, Deep Learning. Please have a seat. Okay, I'll start. Okay, so as we know that there are many different bio networks, and after listening to so many talks on machine learning and AI and deep learning, I wonder maybe HCA should have an AI bio network. So let's start. So let's welcome our first speaker, Wang. He's from Vector Institute, and he will talk about his scGPT foundation model for single-cell genomics. Let's welcome Wang.
All right. Hello, everyone. Today, I'm very honored to present our recent work, scGPT, towards building a foundation model for single-cell multi-omic using generative AI. Before we start, I want to say this is still an ongoing work. We put out the preprint in April. We updated recently. Also, we put the code in GitHub. We welcome your comments and feedback. So what are foundation models? You probably heard it through social media, but academically speaking, This term was coined by a seminal paper from a bunch of Stanford researchers. On this paper I show on the left. And the key idea of foundation model is that we train one gigantic model from massive amount of text such as images. And then once the model is trained, you can take this foundation model and perform fine-tuning on many downstream tasks. So what are the key features of foundation model? And that paper has 200 pages, so too long, don't read. And I summarize as A, B, C, D for the key features of a good foundation model. A, algorithms. Most of foundation models are based on transformers or attention mechanism. Business or applications. The idea of foundation model is that we only train one foundation model that enable multiple tasks by transfer learning. And C, computing, you're going to need specialized hardware such as GPUs or TPUs if you're coming from Google. And last, D, data. Data is the new oil. To train a good foundation model, you're going to need a massive amount of data for pre-training. And the idea we thought about is, can we build a foundation model for single cell data? And the motivation is quite clear, is that while texts are made of words, cells can be characterized by genes. So this analogy really inspired us to explore foundation models such as GPT-like model in single cell. And let's see whether we have all the key features A, B, C, D in single cell. So algorithms, this attention mechanism was published by Google from a paper called Attention is All You Need. And if you stack enough encoders, you will have BERT model. We see examples such as scBERT, scFormer, GeneFormers. These are all BERT model. And if you stack enough decoders, you have GPT-style model. One of the examples is our scGPT. B, business. We have lots of applications in single cell. I list a few in our paper, such as clustering, such as batch correction, multi-omic integration, cell type assignment, perturbation analysis, etc. Computing, as I mentioned, we need lots of GPUs. Thanks to Vector and Computing Canada, we were lucky to have access to some A100 GPUs. Last, data. Again, thanks to the community efforts, particularly Human Cell Atlas, we have access to large amount of single cell data and curated with various organs. So specifically, we took cell by gene census, which contains 33 million cells. I know yesterday they updated, now they have 50 million cells. But we took the one with 33 million cells and we trained our scGPT. And basically, scGPT consists of two steps. First, pre-training, and second, fine-tuning. And specifically for pre-training, we train a model that on cell by gene census, which consists of 33 million. As you can see from this image, that covers multiple organs. And also we show some subsampled UMAP, which kind of shows some oncology of cell types, which proves that the pre-training was successful. Once we have the model, and one kind of quickly brief talk about how we train the scGPT. Specifically, once we have a single cell gene expression matrix, we tokenize the genes first, and then we bing those gene expression values. And more importantly, we can also add condition tokens. For example, which batch this gene expression comes from, or which perturbed genes this perturbation data set comes from. So this condition token give us lots of flexibility to incorporate prior informations. And once we have all the input, the core of the architect is really just a lot of attention layers, which we will not talk about too much. And then I want to briefly talk about how we train it in a generative way. The way we generative train the scGPT is that we use a cell token, which is called a CLS token, which really embeds the cells. And then we look at the attention values of all the genes with respect to these cell tokens. And then we rank all the genes based on the attention values. And then we chop them into different blocks. And then we use the most important block of genes to predict next block of genes. And we also use a technique called teacher forcing, which is a widely used mechanism in NLP, in that particularly at the beginning of the training, we randomly select the true gene expressions from the training data to mimic the generation steps. This was very helpful in the beginning epochs of the training. And once we have a very well trained foundation model, and then the rest is quite easy. Basically, once you have your own model, you fine-tune the base model and then towards your own tasks. Particularly, we talk about how we can fine-tune scGPT for multi-batch integration. Specifically, we use self-supervised loss functions and also adversarial loss functions to remove batch effect. Really, once you define the loss function, the rest is streamlined. You use gradient descent with SGD to train your models. We also compare with many state-of-the-art specialist approach in multi-batch integration. We find that scGPT actually did a better job in terms of removing batch effects while preserving the cell types informations. And similarly, we also perform multi-omic integration. This is an example. We perform fine-tuning of scGPT on paired SC RNA-seq and SC ATAC-seq. Again, we compare with state-of-the-art Serrata V4 and SC glue on this multi-omic integration. And similarly, we did fine-tuning for mosaic integration, in which you don't have paired RNA-seq and ATAC-seq. Again, we compare with the best approach on this particular task, and we find that scGPT can outperform the specialist approach. And then we move to cell type annotation. Specifically, we take 3 data sets and we report multiple metrics. You will find that scGPT consistently outperform the specialist state-of-the-art in cell type assignment. And one interesting aspect we find is called scaling law, in the sense that we train multiple models based on different number of cells. Specifically, we train a model on 30K, 300K, 3 million, 33 million, and you will find that for cell type annotations, the more data you have, the more capacity your model becomes. Although we do see a diminishing gain after we train a model on 3 million cells, but you still see an increase in performance. And then we move to perturbation response. Again, we compare with gears and some other methods. scGPT did a better job in terms of perturbation predictions. And a similar phenomena was also observed in gene regular network. We find that even zero shot, without any fine-tuning, the gene embeddings learned by scGPT can find some interesting gene programs as well. And then we perform some comparison with traditional co-expression pathway analysis. We find that through fine-tuning scGPT, it did find a lot more differential pathways, so in gene regular networks. So this gives us confidence that not only we can learn embeddings of cells, we can also at the same time learn embeddings of genes that can give us meaningful gene program information. Another interesting thing is that we also train many organ-specific foundation models, and we want to see which model is the best to start with. And then we took a COVID-19 data set and to do batch integration, and we find that if you take the base model from blood and the lungs, and it usually gives you the best results. If you take other irrelevant organs, the performance is less satisfactory. If you don't, so this gives us an idea that you should choose the foundation model that is most similar to the data you have. And if you don't have such base model, choose a whole human data set, which we train on 33 million data set. And to make sure this model is accessible to everyone, because not everyone has access to GPUs, we had partnered with AWS and construct this scGPT hub. So it's still on beta testing, but you will find that you basically just sign up an account and then upload your data, and then you choose the base foundation model to start with, based on your own data set, which organ you are looking at. And then you set up some hyperparameters, and then you just submit the job. And then usually for fine-tuning for a data set of, let's say, 20,000 cells, you probably just need to wait for 5 minutes. That's perfect time for grabbing a coffee. All right, so despite some of the benefits, there are lots of limitations for current version of scGPT. Particularly, it's quite limited to RNA-seq. Although we show it can combine with ATAC-seq, we actually don't have a good foundation model for ATAC-seq. It's still not causal. I'm sure scGPT is based on correlations by pattern matching. And also, pre-training is quite compute intensive, although fine-tuning is lightweight. So that's why we built out the scGPT hub to make sure everyone can have access to GPUs. And also a few clarifications. So first of all, scGPT does not use ChatGPT on biological data. And second, scGPT did not find emerging biology yet, although we hoped with more collaborations, with more applications, people can start to find new biology. But so far, we didn't find emerging biology yet. And the scGPT is a foundation model, not so much foundational model that it can solve all the biology. So this is really still computational tools to help people find meanings or embeddings of cells or genes. And moving forward with foundation model is that we hope the whole community can adapt more or less a data-driven approach and require larger scale data with multi-center validations. And more importantly, we want to have a generalist approach with strong support of end-to-end optimized frameworks such as GPU support. And to achieve that, really collaboration is essential. And one thing I want to emphasize using the last slide is we really, although foundation model sounds appealing, we really have to move forward with care. Particularly, there are lots of unanswered questions. For example, how do we curate the pre-training data beyond SC-RNA-seq? How do we evaluate various foundation models? How do we make sure foundation models are open and transparent? We talk about deep learning is black box, but foundation model adds more complexity in interpretability. How do we make sure foundation model is accessible to everyone? And in the end, I want to acknowledge all the contributions are done by two amazing PhDs, and also scGPT Hub was helped by Cornet and Gary as well, and also the whole lab made a contribution too. Thank you very much.
Okay, thanks Bob for the very inspiring talk. So we have 3 minutes for a number of questions. Yes, Li Bo. Yeah, please introduce yourself before you ask the question.
Thank you so much. Hi, my name is Bo from Genentech. Very cool talk. Actually, I have a more like a detailed question. So when you do the data pre-processing, I wonder, do you do highly variable gene selection, or you use all the transcriptome available in the single cell data?
Yeah, that's a very good question. So we can't take all the genes, but because of restrictions in computing, we set the max length to be 4,000, but we can actually adapt to multiple genes. When you do inference, it's better just to select top variable genes, but in theory, we can take all the genes.
Okay, thank you so much.
Yeah.
Yeah, hi, great talk. Michaela from Helmholtz Munich. Yes, I was wondering, I think going in a similar direction as well, but so for one, you kind of evaluate, for example, for the task of batch integration, how good the methods are. I'm just kind of curious, how did you evaluate them? What ground truth did you use, and what is like your opinion also on like the data quality that went into the model?
Yeah.
Did you do any curation for that as well?
Very good question. So we really just took some of the data set that had been widely used by many previous data batch integration methods, and then compare with those data set with so-called ground truths. And most of the ground truths coming from annotations from biologists or some of the differential, like marker gene-based annotations. I don't think it's perfect for evaluation yet, but that's what we can do for now. That's why I was calling, in the end, we really need more rigorous evaluation of various foundation model moving forward.
We have time for one last question.
Thank you for the great talk. My name is Mershad, Gary's a student. So I was wondering, if I generate totally unrealistic simulated data and upload it on scGPT for annotation, what is the output?
A very good question. We actually haven't tested that, but very good question. So I think likely if it's totally random, I think if you draw UMA, it probably will be a bulky either or something indistinguishable. I think the embedding will be similar to the random distribution you generated, but that's actually very good, kind of testing we should do.
Okay, one last question, quick question. Can you meet me halfway?
Hi, Giovanni from Helmholtz Munich. Thank you very much for the great talk. I think it's very exciting the community is looking at alternative ways of learning representation than the classic reconstruction. And in fact, these alternative ways have been explored a lot in other fields and seems to be very powerful in learning semantically preserving representations. So why do you think autoregressive models learn better representations than purely reconstruction-based, specifically considering that the non-sequential nature of the data?
Yeah, excellent point. So actually, before scGPT, we actually published another preprint called scFormer. That is basically kind of reconstruction or random masking-based approach. And the reason we think generative training is more powerful is that during generative training, you have a much difficult task to generate data that fits the data distribution. Therefore, generative training tends to have more reliable fitting to the true underlying data distribution. Certainly, the training will be more difficult, and most of the time, they require more training data. But once it's properly trained, the capacity, most of the time, is better than just reconstruction-based approaches. This is also why you see in NLP side, GPT is more powerful than bird-like models.
Okay, thanks both for the very nice talk and the answers. Yeah, okay. Okay, let's welcome our second speaker, Professor Danilo Bzdok. He's from Milan and the McGill University. He's going to talk about machine learning for single subject prediction. Let's welcome him on board.
Thanks so much. Thanks for the opportunity to talk. So I lead a team at McGill at Milan, Montreal with a mandate to translate machine learning technologies to questions in biomedicine. We mostly study the brain. Disclaimer. So I think I talked to the converted or preached to the converted, but I think it's fair to say that single-cell genomics is this fresh view and this perhaps tighter grip on biology because it's conveniently placed between somewhat static alleles 0, 1, 2 in a big data frame. And it is at the same time easier to analyze potentially than the downstream protein products that cause a lot of challenges. So just with a few subject samples, we can get hundreds of thousands of cells. That's very convenient in a somewhat standardized format. We can do this at scale. It changes the unit of investigation from subjects to cells, which has a lot of advantages, but also some unique challenges. But as a quantitative analyst, my question is, of course, what is the unique structure that we can exploit in this kind of data? And so you know that there's mostly just one kind of linearity, but the moment you deviate from linear structure in data, you really encounter a whole zoo of different kinds of nonlinearity. And I think it's important to kind of keep this in mind. So the final conclusion maybe at the beginning is going to be that single-cell genomics actually has all kinds of amazing nonlinear structure. And so first is just this example here. Just conceptually, you all know MAGIC, which is an imputation tool. Every time you sample a transcriptome, you only capture 5 to 10% of the actual protein transcripts that are in the cell approximately. And we can exploit the naturally existing co-expression networks between genes to impute missing transcripts that we may have missed. But what I want to point your attention to here is really the cell-cell similarity graph step in MAGIC. And because that is where you change the representation and where you apply k-nearest neighbors. K-nearest neighbors in machine learning is known to be one of the most non-parametric algorithms in the sense that you can learn pretty much any form of nonlinearity to the extent that you have enough data. But the kind of nonlinearity that you really capture in this type of technique is what I'm going to call sample-sample nonlinearity. So that isn't really related to nonlinearity that's in the genes or gene expression columns itself. That, in turn, would be an example of t-SNE visualization techniques. Of course, you all know t-SNE in all its variants. But what I want to point your attention to here is that is really attacking nonlinearity from a completely different direction from the top, if you will, if you think about it as an Excel sheet. Because here we really zoom into local neighborhoods of the gene co-expression space. And that gives a lot of advantages, exposes structure that we perhaps couldn't look at otherwise. But it also has this disadvantage, you could call it cursive intrinsic dimensionality, that we cannot really interpret distances between clusters, sizes of clusters. So there's also dependency on hyperparameters. It's a non-convex algorithm, so you don't really know whether you get the same answer in a different lab. It's mostly a visualization tool. You can't really use it for the same things as you could PCA or isomap or something like that. Main point being, this is a completely different kind of nonlinearity again that's not exploited by the method that we had on the previous slide. So there are also hybrids, for example, FATE. FATE is an addition to the t-SNE UMAP visualization stack, and it has the convenient advantage that it uses several kinds of nonlinearity. So I'm just going to call it here local, global nonlinearity. And we probably can learn all sorts of things for all sorts of tasks using these kinds of nonlinearity, independent of what the machine learning algorithm is and independent of the biological goal. So when we look at this single-cell genomics literature with students in my team, we mostly being tourists in this community, we notice that there is a lot of advanced modeling, mostly at the pre-processing stage and at the visualization stage. But what we find surprising is, and that's an opportunity, I would say, when it gets to the actual inference, when it gets to actually isolating a biological phenomenon and saying something about primary biology, I think there's mostly still gene-wise statistical significance testing that's being applied. So I'm not going to say it's the same as what Fisher did in potato crops 100 years ago, but it's not that far from there. And so the question is, can you also use higher order nonlinearity to make inference about biology, and perhaps that's not at the unit of genes, but for example, at the unit of gene co-expression networks that is exploited by the methods I talked about. Nonlinearity is not a given, and I know this from my own experience because I'm going to start with the middle panel here. We conducted the largest existing empirical sample complexity analysis in MRI, more specifically brain imaging. And even the largest existing data sets, if you predict phenotypes of behavior, complex phenotypes, health outcomes, you usually cannot reliably extract nonlinear functions. You cannot really outperform linear models in most cases, in my personal opinion. So in GWAS, there's a somewhat similar situation, I would say. Benjamin Neal's lab published a paper just this year in Science saying that even kind of the most canonical forms of nonlinearity that we should see in common variant genetics, biallelic dominance, and they showed in comprehensive profiling analyses that it's extremely difficult to model this. You would need 20, 30 times the sample size to get the same statistical quality to find a nonlinear effect as you do for the additive effects that are usually studied in GWAS. Or if you take electronic health records, that's a popular theme for grant applications, and there could be a lot of promise, but from my perspective, I have not heard a lot of beautiful success stories about electronic health record modeling. Typically, the moment you look at several variables at the same time, you are very much down in terms of the sample size. So I think a lot of the electronic health records there in the low effective sample size regime, if you really try to do something like deep phenotyping analysis, and that doesn't lend itself to extracting nonlinear structure again. So the conclusion being, single-cell genomics really has very rich nonlinear structure, and that is not a given for, if you look at all sorts of biomedical data. Okay, so now to challenges. So when my colleagues and I noticed that there's always larger data sets, like roughly 10 years ago, we really got excited and we thought we will get always better accuracies with always larger sample sizes in our machine learning prediction models. sadly, that's not how the story went. We observed that the larger our data sets, the more challenging to get good out-of-sample classification accuracies. So that is an observation from the neurosciences. I would submit that that's potentially a similar observation maybe in the single-cell genomics. We do not exactly entirely know why that is. One component, I would say, is as you go away from strict sampling, strict inclusion-exclusion criteria, you sample always broader geographies in a single study, you do retroactive data collection and so on and so forth, you just have always much more different people. And so if you train a model, and that's what you see here, on a general population, but then you apply these models in different ethnicities, you observe that African Americans, AA blue here, those models perform much worse than in white Americans. So that's the case for dozens of phenotypes. For high-dimensional biological data, here it's MRI, but I would expect that that's probably similar in single-cell genomics. So it's not just the prediction performance, it's very different in different segments of human populations. It is also the internal representations that predictive models latch onto. So here you see the similarity varying the degree deliberately of how diverse the training samples are, so the backgrounds of the people in the cohorts, and you can see that you sometimes only have 0.5 Pearson correlation similarity between the internal representations of how a model classifies a disease like autism, for example. So it's the prediction performance and the internal representation. All of this can be quite different if you train different segments of the population. So that is conclusions we came to empirically. This is schematic on the left here. But I think the most concerning is really what you see on the right here. Because oftentimes when you have a biomedical data set, we have age, sex, and then maybe some other socio-demographic or socioeconomic variable. The point of this analysis here on the right is, as you go up in diversity, your prediction performance of the model gets worse. But you cannot really correct for that using standard linear deconfounding techniques, such as linear regression deconfounding and combat. So even worse, and this is single-cell genomic data now from project I'm running with Jack Stanley and my team. So what this plot shows, and I think that's maybe the most concerning, is that no existing batch correction technique actually removes 100% of the batch. So what we did here is, on the x-axis, we apply a certain batch correction techniques, there's usual suspects there. But then we take the same data, the supposedly clean data, and run an out-of-sample standard machine learning prediction paradigm. And on the y-axis, you see that in all scenarios, we can still detect the batch that was supposedly removed. So, and that's the case even if in our embedding visualizations, the batch looks almost the same. So there's, of course, a subtle structure that we can't really observe necessarily at that level. And what we are working on right now, we're not going to try to solve batch correction. I think this is a very challenging problem. Where we try to get a deeper look into what is the batch structure. And so what we do here is explicit modeling of which kinds of batch structure do actually exist in a particular data set. And we can very carefully decompose what is the amount of linear batch structure, what is the amount of sample-sample structure, what is the amount of gene-gene batch structure, and how much do we have of each kind. And better knowing what batch structure you have is, of course, a first step to better explicitly removing batches in your data. And so, okay, I'm going to skip this slide and just summarize with deep nonlinearity in single-cell genomics comes deep responsibility. So we can see deeper into biology in a way we could never before. I'm sure you agree. But I think it also brings things to the surface that we didn't really expect and that will prompt us to come up with new modeling paradigms. Thanks to my partners and funding agencies. So okay. I made a lot of controversial statements, so waiting for the questions here.
Yes. Any questions? Yes.
Thanks. Michaela from Helmholtz. Yeah, very interesting to actually look into the different batch effects. I think that is actually a lot of the time way a more complicated question than you might think in the beginning. I'm curious, so when you talk about, so you mentioned, for example, for integration methods that some of them tend to fail to remove batch effects. What I'm curious about is, like, first of all, how do you define batch? Because I think even defining what a batch is in your data set is also non-trivial. And what are you comparing against in that scenario?
Yeah, so I find it's an interesting research topic precisely because it's not very well posed as a problem. Right? It's somewhat squishy. It's a squishy problem in a way. So I don't think that we encountered a scenario where we actually did remove the batch. That's pretty rare in the data sets we looked at. In terms of what batch, that is I have a very simple answer. We can look at those batches that were actually measured. Right? So you can divide into what are the known unknowns and the unknown unknowns. And you can distinguish between what do you have and what may be a source of variation, but you haven't actually measured it. So the way we attack that is we look at those batch, technical, biological ones, we look at those batch effects that we actually have in the data set. So was acquired several times, there's differences in post-mortem interval, all the things that we actually have. Removing batch structure that is not a part of your data frame is so much more challenging.
So you're saying basically if you don't have that extra covariate or classification in different batches, right? If you say you have multiple batches in one, it's a bit difficult, but like the multiple technical confounders in one, whatever you define as a batch, I would also.
Yeah, exactly. So we're not trying to redefine what a batch is. It's the variation in your data set that is not of primary scientific interest. And oftentimes, empirically, we see that that variation is actually bigger than the variation of interest. And I think somebody said the same yesterday.
And then in terms of interest, so you would say, if you were to consider, say, every donor or every sample as a batch, would you
what
not
sample
be
in this case, person or cell?
Let's stick with donors as a batch.
Donors.
Yeah.
We tried that. That's somewhat, to be honest, it's somewhat more difficult because it's a multi-class classification problem. That's different in terms of like the profile. In principle, we can do that too. So that worked in certain cases, yes.
But it might be removing the exact effect you want to measure, I guess. I don't know.
Exactly. So that is why I think it's interesting to look at the same time. What is the structure of the batch? How much can we remove? How much can we still classify of the batch? And then look at downstream the analysis of interest. What you actually try to do, can you still recover the genes that you think should be distinctive for a disease and so forth?
Please go ahead, yes.
Thanks. Great talk. I just wondered if you try to quantify the amount of the ratio of linear versus nonlinear information.
Yeah, very good question. So actually, yeah, I didn't really say the main motivation for this project, and that is that the deconfounding practices that a lot of biomedical communities has, they are mostly linear still. So single-cell genomics, there's exceptions, I know. For the most part, there's a lot of labs, they just by default do linear batch correction technique. But then downstream, there are all sorts of nonlinear algorithms. So if you do deconfounding for H with a linear algorithm, the nonlinear H-related structure is still in that data set. If you have a downstream model that is going beyond the linear paradigm, you will latch onto that batch structure. And that was the main motivation to actually do these studies. So now back to your question. It depends on the data set. So I think our approach is to actually try to find out in a given data set how much linear batch is in there and how much nonlinear batch is in there. But that has the big disadvantage, as I said at the beginning, there's so many different categories of nonlinear structure, and so you can only test for the nonlinear structure we have a batch correction technique for. So there could still be other nonlinear structure that we just haven't probed yet. So it's data set specific. It's a one-sentence answer.
Okay, let's put our hands together to thank Professor Danilo. Okay, let's welcome our last speaker, Maria Brbic. She is a principal investigator from Swiss Federal Institute of Technology. She's going to talk about deep learning for cell type discovery. Thank you.
Thank you.
Thank you. So this is also my first HCA meeting, so I'm really delighted to be here, and thank you so much for inviting me. So my name is Maria, and I recently joined EPFL as an assistant professor. And today I'll talk about deep learning for cell type discovery. So with the advances of next generation sequencing technologies and efforts of the Human Cell Atlas community, we are heading towards creating these comprehensive reference maps of all human cells. And given this huge amount of data that is being generated, naturally machine learning holds a promise for really transforming biomedical research and becoming the driving force of new discoveries. However, the challenge is that fundamental biology is really different from our prevalent machine learning paradigm. So the first difference is that in biology, we are often faced with kind of collection of small and different data sets that are generated under different experimental conditions. For example, data sets may be generated from different tissues, species, or individuals that suffer from different disease states. However, if you look at our prevalent machine learning paradigm, we are typically assume that there is a massive data set that is generated from a single experimental condition, or in the machine learning language, from a single distribution. And this presents the first major difference between biology and our prevalent machine learning paradigm. The second difference is that in natural science in general and biology in particular, we are really driven by new discoveries. So we want to be able to use machine learning to kind of discover new and unknown phenomena. So for example, we want to be able to discover novel cell types or kind of discover new COVID variants. But if we look at our prevalent machine learning paradigm, what we typically do is that we predict existing and well-characterized phenomena. For example, we assign images to cats and dogs after we see examples of how these animals look like. But natural science is not really about recognizing cats and dogs, but metaphorically saying we want to be able to discover unicorns, meaning new and unknown phenomena we have never observed before. So what biology needs are machine learning methods that can help us to discover these completely new phenomena, as well as machine learning methods that can generalize across different tasks, different domains, different modalities, and so on. And in this talk, I will kind of give a brief overview of some of the methods we developed for addressing these challenges. In particular, in this talk, I will focus on the cell type discovery methods that we've been developing, Mars, Stellar, and most recent method named Saturn. So far, like as a community, we are putting a huge effort to annotate these single cell data sets. And as I said, these data sets are collected from very different experiments. For example, they may originate from very different tissues. And motivated by this huge effort of the community that is put to annotate these data sets, we developed a method that we call Mars. And the key idea in Mars is really to leverage this collection of previously annotated data sets to help us to learn better representation and generalize to a novel unannotated data set for which we don't have any annotations. And these data sets can be very different. For example, they can originate from different tissues. And the key idea in Mars is really kind of to learn to project them into joint low-dimensional embedding space in which we want to force cells to group according to their cell types. And to achieve that, Mars learns this nonlinear mapping function f that projects the high-dimensional input-dimensional gene expression vector to the low-dimensional embedding space, as well as a set of cell type landmarks or kind of cell type representatives in this embedding space. And we want to learn this function f such that cells from same cell types are embedded close together, while different cell types are embedded far away. So I won't go into math details for the interest of the time, and I will just kind of show like a general application of this method. So we apply this method to transfer annotations across different tissues of the mouse aging cell atlas dataset. We also collaborated with researchers from Stanford Biology, Li Chin's lab, and Stephen Quaig's lab to kind of apply Mars to discover different neuronal types across the fly brain development, and also contributed to creating a fly cell atlas, the first single-cell transcriptomic map of the whole fly. And very recently, we also put together aging fly cell atlas where we characterize the aging features of the fly. And while Mars is able to kind of to learn this embedding space that kind of helps us to differentiate these different cell types, the question is kind of how can we automatically determine the cell type identity? So how can we minimize really the human annotation effort put to annotate these data sets? And kind of natural idea that comes first to mind is kind of to utilize some reference label data and then use transfer learning to transfer annotations from one data set to another. And next, I will kind of show an example in which this paradigm simply doesn't work. So you can imagine that you have some reference label data coming from one donor, And in this data set, you were able to kind of label kind of cells to 4 different cell types, and you can train classifier to distinguish between different cell types in a tissue. But now you want to apply this classifier to test data in the wild coming from a new donor, and maybe this new donor kind of, for example, has a cancer, and there are some disease-specific cell types and cell states that you were not able to see and label in your reference label data. So what we ideally want to achieve is that given some reference label data, in this case, for example, if you have label data in which you were able to label cells to 2 different cell types, fibroblasts and cardiomyocytes as a toy example, and then in your unlabeled data, besides these 2 different cell types, you observe some novel cell types and cell states. Ideally, you want to be able to say that these green cells belong to novel cell type 1, these purple cells, you can group them and kind of say they belong to novel cell type 2, and you want to also be able to distinguish between these 2 different novel cell types. So you want to be able to discover these novel cell types as well as identify these existing cell types by assigning them, in this case, to fibroblasts and cardiomyocytes. So to achieve this, standard supervised machine learning paradigm is not sufficient, so we propose kind of an open world semi-supervised learning paradigm, and again, I won't go into math details for the interest of the time, but we kind of design a specialized objective function that allows us to do this. But I will show you next that, how we apply this for annotating the spatially resolved single cell data, which brings us to the method that we call Stellar. So in particular, Stellar takes as input the reference label data coming from one spatial data set. It can be either spatial proteomics or spatial transcriptomics data set. And then we take a data set coming from another donor or a different related tissue. And for this data set, we don't have any annotations. So what we do next is we construct the cell graph based on the spatial proximities of the cells. And then using graph neural networks, we learn the embeddings of the cells that capture both the cell neighborhood similarity as well as the molecular expressions. And then finally, using this open world semi-supervised learning paradigm, we say the Stellar is able to say whether cells belong to the previously seen cell types in the reference label data or discover these novel cell types. And we apply Stellar to transfer annotations from the healthy donor tissue to the cancer donor tissue in a collaboration with Gary Nolan's lab. In particular, this is a data set coming from Codex multiplex imaging technology, and this is the reference label data that we used. And then we apply Stellar to annotate the data coming from esophageal cancer, for which we didn't have any annotations. And these are a result of the Stellar predictions. So Stellar predictions almost perfectly agree with the ground truth annotations, and in this case there were three novel cell types, and this is an example of cyan color shows the one major novel cell type that Stellar was able to discover. And next, we also apply Stellar to annotate the human intestine atlas within the HubMap consortium. So far, Stellar has been used to annotate more than 2.6 million spatially resolved cells. And this is an example of Stellar predictions for a new donor. And this was, again, collaboration with Gary Nolan and Michael Snyder's labs. And while Stellar and Mars kind of operate, in this case, on the same set of genes coming from these different data sets, the next question we ask is kind of, can we go towards universal cell embeddings? Kind of, can we create cell embeddings really for any species, any set of genes? So this is motivated by really the huge effort of the community to kind of construct these to design these cell atlas data sets. For example, there was a Tabula Muris data set, then Fly Cell Atlas, Tabula Sapiens, Lemur Cell Atlas. But the question is kind of how can we analyze these data sets jointly, and can we kind of learn embedding space in which cell types that are conserved across species would be embedded close together, while cell types that are specific for a particular species would kind of form a separate cluster. So besides kind of to analyze these data sets across species jointly, besides kind of standard batch effect challenges that arise when we deal with single cell data sets coming from different technologies, coming from different experiments, there is also a challenge that different genes are really measured in these different data sets. So this brings us to the method, to our latest method that is currently on bioRxiv that we call Saturn. And the idea in Saturn, what Saturn takes as input are these cross-species data sets that are generated, either these can be kind of either cell atlas data sets or maybe organ-specific data sets. And then we use the key idea really is to use the protein language models to encode the meaning of a gene and not only represent genes as a columns in a data set. So in particular in Saturn, Saturn takes these genes across different species, and then we learn to project these genes in the so-called macrogene space, and this macrogene space really represents these groups of functionally related genes. And this allows us to integrate and overcome this heterogeneity of data set across different species, and the similarity of assignments of genes to microgenes is kind of modeling really the similarity of protein embeddings of individual genes. And this is an example of applying Saturn to frog and zebrafish data sets. So this comes on the left. You can see what happens when you try to integrate these methods using standard batch integration methods. So this is just an example of the scanorama, but we also observe similar behavior with other standard methods. And on the right is an example of embedding space produced by Saturn. And this is another example. So this is SAMAP is a really method that was developed particularly for integrating cross-species data sets, while at first SAMAP is able to kind of overcome this heterogeneity and it kind of integrate, and the cells seem to integrate very well. However, if you look at the cell types level, you can see that in the SAMAP case, different cell types often mix together, while in Saturn space, cell types are kind of naturally forming this well-separated space. And this also allows us to kind of formulate, perform really differential expression in the macrogene space, meaning we can ask the question which macrogenes are differentially expressed across species and find kind of groups of functionally similar genes that are co-expressed across different species data sets. And finally, I would like to thank all amazing collaborators that worked with me on this project. And Jana is actually here, and he has a poster on Saturn. So if you want to learn more about the Saturn, please come talk to him today. Thank you so much.
Okay, questions?
Yeah, it looks great. I have a question about the protein embedding. Actually, it's a really cool idea. How do you use actually the information that comes from the protein? Do you use folding information also as well, or just kind of like amino acid sequence?
So this comes from these protein language models that are trained by Facebook, and they are trained really on the amino acid sequence. And they are trained in the similar, you can imagine they're trained basically in the same way as kind of these large language models, so masking some amino acids and kind of predicting the...
So I wonder in that sense if you use like AlphaFold, for example, then because I suspect that between species you can have like proteins that have quite distinct amino acid structures, but the actual 3D structure of the protein is quite similar. So in that sense that you will get like more similarity.
So that would be, I think, a great extension. So you could kind of also, of course, like use maybe kind of extend the method in a sense, because it's also kind of general framework. So you can use any protein embeddings that you want and put it into the method, and method would kind of leverage this information. But key idea really is that we don't represent proteins only as we don't represent genes only as the columns in the matrix, which we standardly do in single cell RNA-seq data, but we're trying to kind of encode some biological properties of genes and leverage that really to overcome this heterogeneity of data sets across different species.
Hi, this is Romain. I wanted to... I had a question, but then another came up. I'll start with the first one. I wanted to know about how you encode the... when you encode the proteins and you have these macrogenes, is it one to one? I mean, one
no,
to...
because
one to many,
genes
but
and proteins, yeah, it's not, but we take the kind of average expression.
I see.
Yeah,
I see.
Because we take the gene, we take the average expression across all the possible protein products.
I see, okay. And then did you think about how to think about, when you have site-seq data, you also measure protein, but the genes encode protein for gene expression. So did you think about imputing missing proteins from, for example, proteomics measurements using some gene expression by using how like the gene expression is actually correlated with the protein and how the genes encode the same protein, and kind of like in silico extending protein measurements, or even modeling the difference between the delay you can have in protein and gene expression, and making models across these modalities by using the protein embeddings as a...
Yes, yes. I mean, that would be super cool. And it's kind of also, as I said, I think it's really general framework. It doesn't have to be necessarily to integrate data across species, right? Because you can also have like a different genes measured because you have like different modalities, right? So you can encode this information into your data set. So far we showed it really on the cross-species data sets, but it is a general framework to think about, I think, to kind of encode some meaning of genes and then go over across modalities, across species, and so on. Yeah. Thank you for the question.
Okay, one last question.
Yeah, thanks. I don't know. Is it working? Okay, great. Yeah, so I have a general question about the cell type predictions. So I'm curious, do you have a way to measure how well your model fits to the annotations, and how do you kind of distinguish between known cell types that you're trying to recover and new cell types? So is there some kind of uncertainty measurement?
So in this case, of course, to evaluate when developing a method, we were evaluating this on the datasets for which we had some ground truth. For example, in this case, for the codex multiplex imaging data, so this dataset was evaluated by the human expert, was annotated first by the human expert, but then when we applied this data in the wild, for example, for this HubMap data, then really we evaluate the method by looking at the marker expressions and then seeing basically whether these marker expressions would agree with kind of the human knowledge, or in this case of this fly brain development, when we were kind of discovering some new neuronal types, then we really kind of went to the biologists and say, like, these are the markers. You can look basically which genes are differentially expressed in the given cluster that you discover, then go to biologists and like check whether these kind of are meaningful, could potentially be kind of meaningful novel cell type that the method finds. So there's, of course, to really interpret the results, there is, of course, we need like some experts to evaluate that these are indeed kind of true novel cell types. Yeah, thank you for the question.
Okay, one more round of applause for Maria. Thank you. Okay, this will end this session. Thank you.
Thank you, everyone. Just wanted to make a few announcements. One is that the breakout sessions are listed here, so CR2, CRC3 in main hall. So please choose one, and we'll go there soon. Afterwards, there'll be a poster session and reception like yesterday. Just a reminder that we couldn't get couldn't get approval to have alcohol in the very public upstairs space where there's a lot of traffic. So just a reminder that the alcohol stays down here. But they're really amazing posters. If you didn't get a chance to see them yesterday, please go there first, and then you can go downstairs to the bar afterwards as a reward. Then we have one more announcement from Ed, who just forgot to make an announcement yesterday in this talk.
Thanks, Gary. Yes, thank you for your attention for one more minute. I apologize that I had meant to make an announcement during my talk yesterday, but forgot to. We are having a computational challenge as part of the Brain Initiative Self-Census Network. This is being led by Bing Ren from UCSD and Joe Ecker from the Salk Institute and the Allen Institute as well. One of the things that's really come from this Brain Initiative work is that we can define the cells with transcriptomics, we can align them across species, so generally good conservation across species, and then we can mine the attack data and find short regulatory elements. And this is really important because these regulatory elements can fit into viruses, they can become vectors for doing very cell type specific perturbations. They could even potentially be used for gene therapy, for precision medicine. It's been very difficult to find these sorts of tools that allow you to target particular cell types, and they can be mined. But at the moment, they have to be mined sort of very manually. This is a very black box sort of thing. You look for peaks that are cell type specific, you screen them out, you find things that can actually do it. We would love for this to become something far more algorithmic, where you can be predictive and maybe even get to regulatory logic. So to set this up, we have generated together a pretty remarkable data set in the motor cortex of the mouse, macaque, marmoset, and human with multi-ohm, with methylation data, with high-C, And we're trying to set up a computational challenge for people to be able to try to come up with the best way to predict these. And we have a big holdout data set where we have actually screened out functional enhancers that we can use to evaluate whether these have been successful. We would love to invite anyone here to participate in this challenge. We're still working out what the prize will be. There will be a prize of some sort. So please, if you're interested, the deadline is early September. So I hope some of you would like to join. Thank you.
Thank you. And I know there are other challenges that people have, so maybe we could disseminate them in the community. So off to the breakout sessions. Thank you, everybody.